All right, thank you very much for giving me the opportunity to talk about the research we've been doing using Blue Waters. I'm Mahmoud Moradi, a postdoc at Taj Khorshid Lab at University of Illinois, and I'm gonna be talking about the transport cycle of a membrane transporter that we have been trying to reconstruct basically at an atomic level, and this is probably the first time that this has been done, which system of this size at an atomic level has been reconstructed in a sense that we have the whole thermodynamic cycle running at the same time, basically on Blue Waters. So first, I'm going to give an introduction on membrane transporters in general, and also GLPT transporter, or glycerol 3-phosphate transporter that we have worked on. Then I'm going to tell you what was that particular thermodynamic cycle that we have reconstructed and studied energetically. The methodology that we use is a combination of a number of different, basically, strategies and approaches. Part of it is some empirical search for good reaction coordinates that we can use to bias the system. And it's based on non-equilibrium simulations and non-equilibrium work calculations. There are also a combination of different path-finding algorithms and free energy calculations that we do in an iterative manner. And at the end, I'm going to show you the reconstructed cycle of GLPT transporter, its free energy profile, and also its global and local conformational changes, and the coupling between these global and local structural changes. So membrane transporters are membrane proteins which actively transport materials across the cell. And they are active in the sense that they pump the substrate against their concentration gradient from a low to a high concentration. And because this is active, they need energy. And the energy can come from different sources. For instance, for this guy, which is called MSBA, it comes from ATP hydrolysis. These transporters are called primary transporters. For some other transporters like these, which are called secondary transporters, the energy comes from the electrochemical gradient of another ion. So the common thing mechanistically for all transporters is what is called alternating access mechanism. It basically says that a transporter has at least two different conformations. One of them is inward facing, which is open towards the intracellular, and basically inside the cell. And we also have another conformation which is outward facing and is open to outside the cell. And the transition happens in a way that the substrate or binding site is never actually exposed to both sides of the membrane at the same time. So this is the general idea about the mechanism of transport in active membrane transporters. But the point is there is a large conformational change which is needed for this mechanism to work. And these larger scale conformational changes are slow, which makes them very difficult to study computationally. Now, the particular system that I have worked on is glycerol 3-phosphate transporter, or GLPT, which is a member of major facilitator superfamily, or MFS, which is the largest superfamily of secondary active transporters. GLPT is crystallized only in one state, which is the inward-facing state. The way that it works in physiological conditions is that the inward-facing state, we have phosphate binding and facilitating the transition to the outward-facing state, and then another phosphate, which is an organic phosphate, glycerol 3-phosphate, replaces that and facilitates the transition back to the inward-facing state. Now, if we don't have the organic phosphate in the system, we only have inorganic phosphate, it is shown that GLPT becomes basically a phosphate-phosphate exchanger. And this makes my job easier because I'm really interested in doing this for one system. I want to basically reconstruct a whole thermodynamic cycle for a transporter. And if I only have phosphate in my system, that makes it easier. So this is what I decided to do. I studied the LPT phosphate complex as a whole, and I tried to see what the transport cycle looks like. So I have basically 4 states: inward-facing apo, outward-facing apo, inward-facing bound, and outward-facing bound. And I want to study the thermodynamics of this cycle here. It is shown actually experimentally using kinetic studies that the barrier for the apo system is very large between the two, and it basically happens very, very slowly. But when there is a phosphate in the system, it brings down the barrier and the transition becomes faster. But the transition between the inward-facing and outward-facing state still is the rate-limiting step. So this is kind of a schematic of the free energy profile of this system that I want to study, but I want to actually calculate these free energies. This is the thermodynamic cycle again, and this is the only thing I have from crystallography. I only have one structure, and I want to basically generate the whole thing. Now, the problem is slow dynamics, as Tom mentioned earlier, that we always have. In this case, it's actually so slow that there's basically no hope that we can do it using just equilibrium simulations. So the regular strategies that are being used, most of them are basically useless here. For instance, running long simulations. This system works on second scale, so we can't even use Anton to do that. Or even multiple copy simulations. Still, as many simulations, independent simulations that we run, we still probably not going to be able to do much. But the enhanced sampling techniques, this is a strategy that may work. And that is basically biasing the system, doing things like that adaptively. But of course, the question is what kind of bias to use and how to limit the conformational sampling of the system. Now, the strategy that we have been using is a combination of multiple copy and enhanced sampling techniques. If you combine them together through something that you can consider it as a loosely coupled multiple copy algorithm, you may be able to actually reconstruct a very slow process like this in a maybe reliable manner. And of course, this is only possible using petascale computers. So first, we started with this part of the process, the inward-facing, outward-facing transition without the presence of the phosphate, which is very, very, very slow in equilibrium, and the free energy barrier is very large. So we tried many different biasing protocols. Here I have a schematic of the approach that we have. First, we try many different biasing protocols and compare them using non-equilibrium work, especially, and other things, until we bring down the work by basically optimizing our bias, or the way that we bias the system, choosing better reaction coordinates, so on and so forth. When we get to a point that non-equilibrium work are reasonable, then we can start doing more accurate pathfinding, basically simulations and free energy calculations. And this is exactly what we did for GLPT transporter. So we used about 100 different protocols to bias this system to go from its known inward-facing state to an unknown outward-facing state. And what happened was we eventually came up with a protocol which seemed reasonable in the sense that we compared this pathway and the outward-facing state to, for instance, the crystal structures from homologs of this system. Or, for instance, we checked if alternating access mechanism is actually followed in the pathway which is generated. And there's several other ways of, I would say, knowledge-based assessment of the quality of the pathway. And we ended up with a kind of simple protocol at the end. We found two particular helices that if you rotate them, you basically get the other state. So after doing this, then we can do some pathfinding algorithm. We can optimize this pathway even more using pathfinding algorithms. So I have a schematic of the type of pathfinding algorithms that we use. A particular class of these algorithms is called string method. In this approach, you usually start with a string of different conformations, or basically a number of images of nodes representing a pathway in a high-dimensional space. Then you update these centers in an iterative manner, for instance, to get to the minimum free energy pathway. Usually it involves some sort of a reparameterization, so on and so forth, to make sure that these are covering the entire pathway. A particular pathfinding algorithm that we have used is string method with swarms of trajectories, which is something which Blue Waters resources making it possible for us to do. The parallel version of this algorithm basically requires usually something like 1,000 or even more replicas of the system running simultaneously and talking to each other. So in this algorithm, you have a number of different structures for each node, which you run very short simulations like MD steps. And at the end of that very short simulation, you calculate the current position. And then you restrain that to that new center for a very short time, and you release that again, so on and so forth. So this is a loosely coupled multiple copy algorithm, for instance. So these independent simulations are running, but they are also talking to each other occasionally. Once we have the pathway, which is refined using this method, for instance, then we can do free energy calculations. For free energy calculations, we use another loosely coupled multiple copy algorithm, which is called bias exchange umbrella sampling, which is a combination of umbrella sampling, which is a very well-known free energy calculation method, and replica exchange method. So the umbrella sampling method is basically based on restraining the system along your reaction coordinate or pathway. Now, if you restrain the system along the pathway in multiple copies, what happens is you basically sample the entire pathway. Now, there is a problem. We don't have diffusion along the pathway. To solve that problem, you add this replica exchange element to it, which allows basically these different replicas exchange with each other information. And in some sense, you can think of it as kind of a free diffusion along the reaction coordinate if it's designed correctly. So one more element that we added to this to these two algorithms to make it iterative is what I call post hoc string method. So if you do, for instance, free energy calculations using this bias exchange umbrella sampling, you get a lot of different conformations. This algorithm is an analysis method which basically extracts the best representatives of a minimum free energy pathway and then passes that to string method with swarms of trajectory, which is a multiple copy algorithm. And then you can iterate basically over this until you converge both in terms of the quality of the pathway that you get and also in terms of the free energies that you obtain. So using this method, we basically studied this leg of the cycle first, and then we also did the other 4 legs, and eventually we had the 4 legs basically reconstructed and characterized, and then at the end we put together all the conformations that we get from all these pathways and generate a kind of a one-dimensional curve, a closed one-dimensional curve in a high-dimensional space. So these are some of the simulations that we ran to get to this point. So some bias exchange and string method simulations for APO case and so on and so forth for the other legs. Sometimes we, for instance, needed to run 2-dimensional free energy calculations, which requires, for instance, 200 replicas. Our string method calculations required 1,000 replicas. These are all running simultaneously. And at the end, we basically put together 150 images of the system or conformations of the system and ran a bias exchange umbrella sampling. They are basically put along this one-dimensional curve at different points, and they are exchanging information between each other, and they are kind of sampling the entire pathway. And that allows us to actually calculate a free energy profile, which is basically hysteresis free. So this also shows the scheme that we used for this calculation. Started from the only crystal structure and ending up with the full cycle. Using this method, we basically ended up reconstructing the entire cycle at an atomic level and also the free energy, which kind of looks like what the schematic suggested. But now we have actual numbers here that we can compare. For instance, how much the barrier comes down, which according to our calculations is about 3.5 kcal per mole. The barrier comes down when you go from an APO system to a phosphate-bound system. We can also look at the role of different helices in closing, opening of one side and the other side, so on and so forth. One very interesting observation that we made and wasn't suggested before for transporters is the fact that we observed the conformational transition pathway of the protein changes when the substrate is actually present and when it's not present. So the way that the substrate brings down the free energy barrier is by changing the conformational transition pathway. We can also look at different residues. We have all the chemical details. We can see what different residues do, what is the role of different salt bridges, what is the role of different residues in binding the phosphate. And what is very interesting is that the phosphate is bound in the outward facing case very differently as compared to the inward facing case. So we have conformational dynamics in the binding site. And this is my final slide, which I'm going to show you about this research, which kind of summarizes what we have done. We have the full cycle here shown, and also we can see how different residues are involved in binding, so on and so forth. And also we can look at the pore radius, which shows us how alternating access mechanism works. We have the free energy, and this basically summarizes my work. At the end, I would like to also mention that this method was developed basically in this way, combining all these different elements to solve this particular problem. But now this is an established method that we can use it for many other systems, not just transporters, but definitely starting with other transporters. And this is exactly what we are doing. Several people in Taj Khorshid lab are now working on many different transporters to use this approach to also reconstruct their thermodynamic cycles. So I would like to thank my advisor, Professor Taj Khorshid, and Dr. Ankawi, who helped me in this project, as well as other people in the lab, and also the funding agencies. And we did the shorter simulations using XSEED, but most of these large simulations, I mean, all of these large simulations, which require hundreds of replicas, they are all done using Blue Waters. Thank you very much.
So how do you actually communicate between the replicas? What method do you use to communicate the information?
Right. So actually, this is something that maybe I should have mentioned, that basically what they did in NAMD, and I wasn't part of it, they implemented this in NAMD, which using MPI, allowing the different replicas to talk to each other, and they made it available to us as practitioners to use TCL scripting to manage that. So we just do TCL scripting using certain MPI commands that they have generated for us, that now we can use whatever algorithm that we like to have them communicate with each other.